My project is diagnosis of depression disorder using mRNA approach. Major depressive disorder is a chronic mental disorder caused by stress, events, illness, etc. Here's a few symptoms and diagnostics. This experiment will explore genes and biomarkers correlated with depression to reject or fail to reject the null hypothesis. These are the tools I used. With this chosen dataset, I did 3 comparisons with GEO2R: depression versus healthy, remission versus healthy, and depression versus remission. Only the depression versus control graph depicts genes with p-values under 0.05, meaning they're statistically significant. The UMAPs show clear separations in depression versus control and depression versus remission groups. Overall, only depression versus control comparison had 2 separated groups with many significant p-values, so I continued further research solely on that comparison. The heat map shows the expression levels of each gene per sample. Most genes suppressed in the control samples are highly expressed in depression samples and vice versa. I kept the top 200 up and down regulated protein coding genes with p-values under 0.05. Looking at both string maps, the upregulated genes have more enriched pathways, which I continued analyzing. The pathways correspond to gene ontology terms, ancestry charts that help better understand cellular functions. I selected the top 4 pathways shown in this column. The color-coded genes correspond to the selected pathways. The chances of no difference between test and control was negligible for 5,000 significantly expressed genes. Based on some enriched genes functions, there is upregulation of stress-responsive proteins, glycoproteins for blood coagulation, just to name a few. Hence, I reject the null hypothesis because of the evident differences.